Good morning, everybody. This is the WIGL Summit 2, The Importance of Biology in Managing Woody Species. I'm Mari Hardell, and I work with the Minnesota Department of Agriculture as a noxious weed specialist for northern Minnesota. I'm at home today in Duluth, Minnesota, and wish we could have hosted you here for an in-person conference, but I think this virtual conference is working out pretty well. As the session goes on, you can type questions into the question and answer box, and then at the end of the 4 speakers, we'll have about 20 minutes to go through those questions. But we'll go ahead with Theresa, and Theresa Culley works with the University of Cincinnati, and she'll talk about the biology of Callery pear and why it is so difficult to eradicate.
All right, thank you, Mari. So as she mentioned, I'm from the University of Cincinnati. I also work with the Ohio Invasive Plants Council on our assessment team, and I'm also a member of the Midwest Invasive Plant Network, so things that I really enjoy doing. But today we're going to be talking about the Callery pear. I'm actually going to be just continuing on from what Lauren was talking about. This is a great example of a lot of the things that she already mentioned, so she set it up really nicely. If you've heard me talk before, the first few slides will be familiar, and then we're going to get into some new information. So many of us are familiar with the Callery pear planted around our areas, but a lot of people don't realize that it's exactly the same thing as when we mean Callery pear. We're talking about the Bradford, the Cleveland Select, the Chanticleer, the Aristocrat. There's a whole bunch of different cultivars. But the question, and for me as a scientist, I love curious, weird things. The question is, how did this tree that's been in our landscape for a number of different decades, how did it become essentially this? So this is a case where we have a large, extensive, invasive population in Butler County, right off the 75 freeway, right near where I live, north of Cincinnati. So that's what we'll be talking about today. If we look at the distribution of the species, essentially what's happening is it's moving northward and westward. And so we're starting to see, as you can see in this map here, different counties where it has been found. And there's more counties that haven't made it on the list. For example, I've seen it in Reno, Nevada. Interestingly, the Callery pear, Pyrus calleryana, is facing, as it says here, increasing regulation. It is starting to appear on many state-based lists, and you can see it here. In the orange is where it's been listed as invasive in states. Orange is it's on the watch list, so people are keeping an eye on that. Green obviously is it's not listed, and this lighter green is an interesting case. Illinois is the only case here where it has been recognized as being invasive, but it has been removed and not listed as invasive. In this case, there's concern with the economic impact. This is also valid for many other states, as you can see below. And the economic impact, of course, is due to the fact that it's a very valuable landscaping tree, an ornamental tree. So essentially, the Callery pear, it's a really fascinating story. This was originally from China. It's where it's native to. It was brought over into the United States in the early 1900s, and it was brought over for several reasons. The primary one, though, is at that time, there were huge plantations, if you will, populations of Pyrus communis that were planted on the West Coast. And this is the edible pear. So the picture that we show here. These plants were unfortunately falling victim to fire blight. And so it was just decimating large groups, swaths of these planted trees. And so the USDA wanted to come up with a way to breed fire blight resistance into Pyrus communis. So they sent plant explorers to China to try to find a fire blight resistant related species, and they found that in Pyrus communis. The Bradford was eventually, again, grown. It was in 2 different areas of the country, one on the East Coast in Glendale, Maryland, and on the West Coast in Corvallis, Oregon. Now, in Glendale, Maryland, after a few years, one of the researchers there at the USDA Agricultural Station noticed that there was a pretty attractive plant that was growing from seeds that were brought back from China. And he thought that this might actually be attractive enough to be desirable as an ornamental tree. And so this is how the Bradford got started. The Bradford, he cloned the tree. Eventually it was named Bradford. And this is an example of where it was planted out in the residential areas at Glendale, Maryland. This is in the 1960s about 1961. It was then put full blown into commercial propagation, and it became very popular, as we know, sold all over the place. However, it does have one flaw, and it's actually a fatal one, is that the branching structure is such that there's not a central leader of the Bradford. And so essentially over time, after about 15 to 20 years, if you have a windstorm, an ice storm or something, many times the branches will start to break apart. In this example here, this was after Hurricane Ike came through my neighborhood several years ago. This is what happened. And so the trees were essentially split. Now, people love this tree, and they really wanted to have something that was more long-lived that they could have in their yards. And so that's why these different cultivated varieties or cultivars came about of Pyrus calleryana. Shown here are just a few examples. Of course, we have the Bradford over here, the original one. You can see the Aristocrat, one of my favorites, Cleveland Select. Capital is more of a column structure, Red Spire, Autumn Blaze, obviously gorgeous red color. And there's a variety of different types of cultivars published or available today. Now, originally, we didn't have any seeds produced, any fruits produced. And shown here in the bottom right is an example of what we're talking about, these fruits. They're not the edible Pyrus communis fruits, but these are the smaller Pyrus calleryana. Now, it had been suggested that maybe these fruits are now starting to appear because these different cultivars are cross-pollinating. And so that is something that my lab decided to look at. Now, the story is also a little bit complicated, but yet more interesting because of how these cultivars are propagated. So they are propagated by grafting. Essentially what it is they are cloned from a single individual. So all Bradford trees all originally came from one single tree, one single genotype, the Bradford, that was initially at that Glendale, Maryland station. Now what you do is you take cuttings of that tree, it's called the scion, and then you graft that onto rootstock. The rootstock is usually other Pyrus calleryana trees or saplings that have been cut, and then you just essentially put the two together, align them, tape them together, and if you're lucky, it will grow right into it. So here over on the right, this is a tree that I purchased from Home Depot many years ago, and you can see the graft point right here. So anything north of that point would be the scion, anything south of that point would be the rootstock. So the point here is that an individual tree that you buy in a store is actually composed of two different individuals that have been grafted together. So essentially, that's just what I said. All the top part of the trees of each cultivar are all genetically identical. So all Bradfords are genetically identical, but each one, each tree, the top part and the bottom part are different. This is going to be very important for our study. So initially, we wanted to know where do all these wild populations of the pear come from? Starting in the early 2000s, and even before that, in some areas of the country, we started to see these wild populations popping up along the roadway. People didn't really know where they came from, but they had some indication. There was some information being shared on gardening forums on the internet saying, you know, maybe this could be the cultivars mixing together. And so that is what my lab decided to study. So our first step was we did a hand-pollination study. So we got individual trees purchased from different stores around, and essentially we took individual flowers and we emasculated them. So we pulled off the male portions and then pollinated those using pollen from either the same tree, a different tree of the same cultivar, which technically should be just the same, right? Because the top parts are all genetically identical. Then we use a different cultivar to try to see if we might get fruit set with a different one. And then just to make it interesting, we grabbed pollen from a wild population. Shown on the right here is a typical example. This is just using the Bradford as a maternal parent. We used two other cultivars as well, got the same results. Here's an example of percent fruit set on our y-axis. You could see that when we did pollinations within the same tree, or we used pollen from a different Bradford, a different tree, but still the Bradford, you can see 0% fruit set. However, you have 100% fruit set if you then use pollen from 2 other cultivars, and then you get very close to 100% fruit set if you use pollen from 2 wild populations, the Benedict site that you'll see in just a moment, and then we had a site near one of the freeways. So this is showing us that yes, people were actually correct when they suggested that fruit set is being caused by pollination, cross-pollination among different cultivars. So we wanted to go a step further. So we tested that with genetic markers. So we wanted to compare the DNA of our wild trees to the DNA of known cultivars, essentially to try to figure out who the parents are. So it's a pretty simple procedure. It does take time though. You collect the leaf, you take it to the lab, you extract the DNA. The DNA essentially looks like a bunch of snot in a tube. And then using a polymerase chain reaction in the lab, a special reaction where you can amplify very specific parts of the DNA and compare them, you could figure out what trees are related to what, or which ones are the same ones. And so that's what we did. So here's an example of what we found. And so in this case here, each of these columns is a single tree. And this is called, it's genetic results, and it's run through what's a program called Structure. The important part though, is I want you to pay attention to is, again, each column is a separate tree. And then the program will color code different trees that have the same identical genetic signature. And on the bottom here is the name of the tree that it was labeled when we went and collected it. So these were all trees from local stores in the area in Cincinnati. You could see, first of all, that all trees that were being sold as Aristocrat do come out exactly the same, 100% of this yellow color. We go over to Bradford, and you could see that most of them are genetically the same, this blue color. However, we have one case here, it's red. This is actually a red spire tree being sold as a Bradford. And here you could see 3 Aristocrat trees being sold as a Bradford. And red spire, similar type of thing. You could see there's 2 interlopers there who are not the right type. Capital, they're all the same. And then something really interesting happened here. Before we get to that, I just want to mention, so what we found for the most part are that individuals within a cultivar are genetically identical, but I pointed out those exceptions. Turns out that some cultivars are sold mislabeled, and it's about 11 to 12% of trees that are sold mislabeled. So you might be thinking you're buying a Bradford, but you're actually buying something else. Now, what I was getting at is that you could see that a number of different cultivars came out as genetically identical. The Chanticleer, Cleveland Select, Stonehill, and Fari. At first, we thought this was a mistake. We thought we had swapped samples somehow. We ran it 3 times, got the same results, and started asking around. It turns out when I started talking to some nursery folks, they said, oh, this makes perfect sense because everyone knows but me, that we had 3 different cases where a plant breeder or a developer was driving through some area in Cleveland, Ohio, and saw a gorgeous tree and thought, wow, I should clone that. That could be a wonderful cultivar to sell. And so that happened 3 times. That resulted in the Chanticleer, the Cleveland Select, and the Stonehill. Again, apparently these are all clones of exactly the same tree in Cleveland, Ohio. And so that makes sense. So the message that I got is you can go to Home Depot and buy Cleveland Select for $10, or you can go somewhere else and buy a Chanticleer for $100, but it's the same thing. So using this genetic information, we're able to do really cool things. So this is an example, an overview of the Harris Benedict Nature Preserve in Cincinnati, Ohio. This is a preserve owned and managed by the University of Cincinnati, of course, where I work. If you're familiar with this area, this is the I-71 freeway off to the west. This is Sycamore High School. This is Cornell Road, and along the top here is the 275 Outer Belt. One thing you'll notice is half of this preserve looks a little different than the rest, and it is. In 1999, a tornado came through and pushed out, damaged a lot of the area, pushed down all the trees. Many houses were demolished. Unfortunately, several individuals were tragically killed. And what happened though, this gets back to sort of what Lauren was talking about disturbance, is that it was obviously a major disturbance. And you could see here all these wild individuals, these trees started to pop up. These happened to be along fire lines. But using genetic techniques, we're able to sample these wild trees, determine their parentage, and then you can see also go all into the neighborhood and sample trees in people's yards and figure out what they are. So you can see most of the trees in people's yards in this particular area are green, so they're Bradfords. You can see we have a few aristocrats, we have some red spires, and then when you look at the wild offspring, most of them are F1 hybrids, and you can see most of them are Christmas colors, so red and green. And so, in other words, they're an F1 hybrid between a Bradford and aristocrat. So that's interesting. So that sort of confirms the suggestion that people had a while ago that maybe the different cultivars are getting together and crossing, producing fruits, which are then being carried off. But we started to think about this a little bit more. And remember, I told you how these individual trees are propagated through grafting. So we started to realize that perhaps crossing can occur between the rootstock and the upper scion. And this is possible. This is a tree I was driving by one day near the university, and it looked interesting. So I got out and I checked. And when you look at the center of the tree, you could actually see the main stump had been cut, and then the rootstock had actually grown up. So what you're thinking is a large bushy tree is actually rootstock, all flowering like crazy. So what we did was we sampled the scion and rootstock of 57 trees in southwestern Ohio. Then we looked at them genetically. It turns out that they are always genetically different. So the upper part and the lower part of every tree, without question, were always different from one another. So this suggests that now you could potentially have a wild population form from a single cultivated tree if the rootstock is allowed to grow and flower and it crosses with the upper part, the scion. So that's important. So in summary of this portion, what I want you to remember is that these wild pears, the wild populations, are formed in the following way. We have several different cultivars that might be planted within an area, often, around people's houses. We have, I haven't told you, generalist pollinators that love these things. They fly back and forth, pollinating like crazy. Because of that, we have these fruits that developed. And the other part that's pretty important that gets back to what Lauren was talking about is that these fruits are bird dispersed by the European starling primarily. And we'll come back to that in a moment. And so the starling just flies everywhere and defecates the fruits after they consume them, and thereby dispersing the seeds. So we also mentioned the upper part, the scion, can cross with the rootstock, and same thing can happen. Those fruits get out into natural areas. So in summary, wild populations are originating from cross-pollinations in two ways: between genetically distinct cultivars, also between the scion and the rootstock. Okay, so that's interesting, but what can we do with that? And more importantly, how do we control this thing, right? So we can ask the question, how readily can wild pear seeds and seedlings establish? And so this in my hand here on the left is a whole bunch of pear fruits later on in the winter. These usually hang on because the birds don't really apparently like them so much, and will go for other things. Eventually, they'll start consuming these fruits. To get the seeds, you actually have to get vice grips and crack them and get the seeds out. It's actually quite difficult to do. And so we did that though, and we wanted to compare seeds from cultivated and wild trees. And we did this from six different sites. Now, if you think about it, seeds from a planted tree in someone's yard are going to be F1 hybrids. If it's a wild tree, which could already be an F1 hybrid or F2 or a backcross, it could be something else. So we just call those advanced hybrids. This is the results that we found. This is based on percent seed viability, and I've separated this here by cultivars. The main point is that regardless of whatever the parent is, the cultivar type, this is the example of the cultivars, seed viability is pretty good. It might be a little less than 40%, but it can go all the way up to 80%. Capital is one of the best you could see. Interesting thing here is that Chanticleer, Cleveland Select, Fari, and Stonehill all are genetically or are statistically similar to one another. That's why they all share the C number or letter. And this is totally consistent with what we found out before, in that these are all exactly the same plant, essentially. So we also look
Theresa, you have 1 more minute.
Thank you. And seedling survival, and that's what we found. So, and then even 11 years later, that is what we had here. And you can see it hasn't changed at all. So it suggests essentially that wild populations have a seed bank and continued control will be necessary. So I'm just going to give you one more thing to think about here, and then we'll stop there for questions. And that's, are wild pears always found in open and edge habitats? It turns out that no, they're popping up in forest areas. And we wanted to know, is it a certain type of hybrid adapted to the forest understory? The answer is no, it's not. And so what's going on? And so what I want to go back to what Lauren was talking about, it comes down to the birds. Previously, the European starling was the one dispersing it along the edgeways and the roadways. Now what we're seeing is, they're still doing it, but the American robin is now apparently taking the seeds, and they have a different flight pattern where they fly into the interior part of the forest. So I'm going to go ahead and stop there since I'm nearly out of time. If anyone's interested, we can, I can send you the slides about various steps to stop the wild pear. One thing I do want to mention is that we do have a new sterile cultivar that has just been produced called Chastity. And so that is an option that might be available. So thank you very much.
Thanks, Theresa. And Theresa will have to step off before our question and answer session. So we'll go ahead and ask a couple of questions now. So someone is wondering if your research findings on the 3 cultivars being genetically identical have any economic impact.
Oh, well, not really in terms of invasiveness. I mean, they would have an economic impact because if someone were to buy one of the more expensive trees, you know, it's the same as the less expensive tree. So it would have an economic impact in terms of someone's wallet. In terms of impact on the invasiveness, no, I mean, they're all equally invasive.
Sure. And then do you happen to have a map of the worst areas of spread in Ohio?
Oh, good question. I don't, but we should. I'm trying to think if we do. I could tell you that it's been primarily moving north, but it's centers of it's going to be all over. Columbus was where there's a huge spread, of course, in Cincinnati, moving northward. Yeah, but we could definitely get that. That's something good that we should have.
Thanks. So we'll go ahead and move on to our next presenter. And so next up is Dave Gorchov with Miami University in Ohio. And we will learn about the interactions between the invasive Amur honeysuckle and overabundant white-tailed deer and the implications for management.
Thank you very much. Hoping that the PowerPoint is visible to everyone here.
Looks good.
Give you a little video of an overabundant white-tailed deer browsing on Amur honeysuckle in southwestern Ohio. Some brief background. Amur honeysuckle, Lonicera maackii, is from Northeast Asia, the Russian Far East, Japan, Korea, several provinces in China. It's widespread in many parts of the US, including the Midwest, and I may grab the FIA map that I learned about this morning to replace this one. And it's one of these shrubs that has the extended leaf phenology of expanding leaves early and retaining them late in the fall. We'll see that becomes important for its interaction with deer. My early work with this species was looking at impacts on native plant species. I'm just going to show you one slide focusing on effect of tree seedlings. Where we've removed honeysuckle, we have higher survival, higher growth of native tree seedlings. But we also have white-tailed deer increasing across many areas of North America, very dramatic in Ohio in particular. And the negative effects of deer on native plants have been well documented, particularly their effect on tree seedlings, which will be my emphasis today. I'm just going to show you data from one paper from the Midwest, from northern Wisconsin, Bradshaw and Waller. They used FIA plot data. They looked at the abundance of saplings of different tree species that they had sorted into different palatability groups, palatability to white-tailed deer. And they looked at the abundance of these in each plot each decade, comparing it to an estimate of deer density the previous decade from the local wildlife management district. And the main point here, let me grab my little pen tool. So species that were more palatable in these groups 3 and 4 here were very sparse. There were very few saplings anywhere attributed in this study to past high deer density. This intermediate category, where deer were sparse, saplings were abundant, and where deer were more abundant, saplings were sparse. So the deer are not just browsing on seedlings, but they're preventing seedlings from reaching the sapling stage and on into the canopy. Just as an aside, seed dispersal, deer as well as some bird species disperse viable seeds of Amur honeysuckle. This is work of Steve Castellano and Pete Guyden. But we're going to focus on what the combined and interactive effects of these two stressors are to native plants. And this is data largely drawn from a field experiment we started in 2010, where we established deer exclosures, 20 by 20 meters. And each exclosure was paired by a nearby plot that had deer access. And in half of each of those plots, we removed the honeysuckle, which was by far the most abundant shrub in this whole area. And the area was the natural areas near Miami University. On the bottom left, you can see what one of these exclosures looks like. It's an area of high honeysuckle abundance and also high deer abundance. This work was started by Jessica Peebles-Spencer as her dissertation and continued by Christina Hafey, an undergrad in my lab. And we started off with this understanding that honeysuckle itself has a negative effect. So if you compare a plot where we've removed honeysuckle to one where the honeysuckle is still present, we have lower performance of some plant community parameter. This might be species richness of native plants, for example. And this might be the same both where deer have presence and where deer are excluded. The deer, in turn, have a negative effect, again, from our studies and the literature. And whether or not honeysuckle's been removed or honeysuckle is present, we would expect there to be some negative effect of deer. If you get these parallel lines in this mean performance of whatever our response is, this would be an additive response. But it's also possible the response is synergistic, meaning areas that both have deer present and have honeysuckle present would perform even worse than if you added together the negative effects of each. Or the effects could be subadditive, where the combined effect, where deer are present and honeysuckle is present, might be less bad than what you'd expect from adding the two effects. So these are the three different predictions under additive, synergistic, and subadditive effects. And most of the graphs I'll show you will be in this format. So the first is on total cover of native forest floor plants. And so we're going to focus on this panel here of native cover. And what we see is the subadditive response. High cover of native plants where deer are excluded and honeysuckle's been removed. And pretty similar in the other three treatments. And we would see the same pattern if we look just at cover of spring perennial herbs. However, for the species richness of tree seedlings, we only see a deer effect. So where deer have access, we have lower species richness per plot of trees than where deer are excluded. No honeysuckle effect. And if we looked at tree seedling stem density, we'd have the same pattern, and for native shrub basal area. So a deer effect, but not a honeysuckle effect. But we're back to this subadditive response when we look at percent cover of tree seedlings. This says tree cover, but this is in the forest floor layer. Again, high cover where deer are excluded and honeysuckle's been removed, and pretty similar for the other three treatments. So this subadditive response matching this prediction. So this is very common response. So why do we have this subadditive effects? And I think there's two reasons. The first is honeysuckle actually mitigates the negative effect of deer. And the second is that deer actually mitigate the negative effect of honeysuckle. We'll take these two in turn. First, honeysuckle mitigating the negative effect of deer. If we look at some data on seedlings per square meter, only looking in the deer access plots, if we remove honeysuckle, we often have lower seedling density. So these are our 4 different study areas. Let's just look at these blue bars, one study area. Where we've left the honeysuckle present, we have 0.1 seedlings per square meter. Where we've removed the honeysuckle, we have less than 0.03 seedlings per square meter. So removing honeysuckle reduces the seedling density. And the other way to think of this is the presence of honeysuckle is mitigating the negative effect that deer have. And that was tested explicitly in this experiment that Jessica Peebles-Spencer did in the same general area where we had our deer exclosures, but using these small exclosures, you may be able to see the netting here, that are built around large isolated honeysuckle shrubs. And we planted tree seedlings under the shrubs and just half a meter away from the crown of the shrubs. And we did this inside those exclosures, represented by this dotted line, and in other large shrubs in the same area where we didn't put exclosures. So we can look at the effect of being under a shrub, honeysuckle shrub, with deer present or deer absent. And at least for sugar maple, we had this facilitation effect. If we compare just where deer have access, so this light gray color, survival of sugar maple seedlings was high underneath the honeysuckle shrubs and low away from the shrubs. And we could see browse damage and so on that accounted for this difference in survival. So the presence of honeysuckle facilitated survival of sugar maple where deer were present. Now, where we excluded deer, it was the opposite effect. The sugar maples had higher survival when they weren't under the shade of the honeysuckle shrubs. So, so the honeysuckle is somehow protecting sugar maple seedlings from deer browse. Now, we didn't have that effect for shagbark hickory and white oak. These seem to be so preferred by deer that the deer will find them and pursue them, even if they're a honeysuckle shrub. Now let's look at the converse. What's the evidence that deer are mitigating the negative effect of honeysuckle? Well, pretty early on, we noticed that deer were actually browsing honeysuckle. Now, initially, we were surprised because we think of invasive species as benefiting from this escape hypothesis, that there are specialist herbivores and pathogens in the native range of these species that are keeping the species in check. But when we bring them to the United States, they are lacking those specialist herbivores and pathogens, and they can have higher survival and growth. That hypothesis is focused on specialist enemies. White-tailed deer are generalists and have not read about the escape hypothesis. So the first thing we did is actually look at the cover of honeysuckle back in our exclosure experiment. And so here we're comparing those half plots where we didn't remove honeysuckle. We just left it alone, came back 4 years later, and compared where deer had access versus where deer were excluded. And this is a cover of honeysuckle in 3 different height classes, 0 to half a meter to 1 meter, and 1 to 1.5 meters. And in these 2 higher classes, we have lower cover of honeysuckle where deer have access, higher cover inside the exclosures. So this is evidence that deer are browsing at least in this range of half a meter to 1.5 meters of the honeysuckle shrubs. And this actually affects the diameter growth of these honeysuckle shrubs. So again, we're comparing deer access plots and inside the exclosures. And now we're looking at the basal area of honeysuckle, measuring every single stem and comparing it to how big they were in 2000, so over 5 years. And in both plots, there was more honeysuckle, so honeysuckle had grown, but it grew much more where deer were excluded. So the presence of deer wasn't eliminating the honeysuckle, but it was slowing the growth. So deer are mitigating the shading effect of honeysuckle. And so this raised the question of why deer eat honeysuckle. And Ellie Wright tested 3 different hypotheses. The first is, this is actually a preferred species and deer are browsing it because it's preferred. The second is it's not preferred, but where the other species that are preferred, oaks, hickories, maples, and so on, where those have been depleted due to high deer abundance for decades, then it's eaten as a food that's still available. And the third is that it's a unique food resource. It's not really something that's just ranked with other foods, but it plays a different role in the annual diet of deer. And what we think in particular, that during a season of food scarcity, and just a little hint, this is going to be late winter, early spring, it provides a nutritious food. So we tested this in 8 sites in southwestern Ohio, where Ellie very carefully compared the proportion of twigs of honeysuckle that were browsed with the proportion of twigs of each of the native woody species within the height range that deer browse woody plants. So the first hypothesis was rejected. What I'm showing you here are electivity values. Electivity is the extent to which something is utilized based on its availability. So a 0, which we did get for one of our sites, indicates no preference, that the consumption of honeysuckle in this case was just in direct proportion to its availability in that browse range. A negative number, which most of these sites has, indicates that it's avoided. There's still some consumption of it, but it's less than you'd expect based on the abundance. And a positive number, which we did have at one site, indicates it's browsed at greater frequency than you'd expect based on abundance. But at most sites, the electivity is negative, meaning it's avoided. It's not a preferred food for deer. We had some equivocal support for the second hypothesis, that deer are eating honeysuckle in places where the preferred species have been depleted. So at each of our sites, we calculated how many twigs per square meter there were of these more preferred species. And these were species that consistently had higher percent browse than honeysuckle did, again, within the height range that deer can reach. And here's our electivity value of honeysuckle. And again, a negative value means avoided, deer avoiding honeysuckle. A positive value meaning deer prefer honeysuckle. So there's some trend, but it's not significant, where there are very few twigs of these more preferred species, then honeysuckle becomes a preferred food. But the hypothesis that really had support was the third hypothesis, that honeysuckle provides some unique food resource, which again, we think is a nutritious food during a season of scarcity. And the reason we favor this hypothesis is this negative relationship here, that the proportion of honeysuckle twigs that were browsed was highest where honeysuckle is sparse. So here's a measure of honeysuckle twigs per 100 square meters. And where honeysuckle is sparse in the landscape, the proportion that's browsed is really high. And where honeysuckle is common in the landscape, the proportion browsed is low. So it suggests there's like this constant mass of honeysuckle twigs that deer are going to eat, regardless of what else is out there. And this is what would happen if there was one time of year when all the deer were eating honeysuckle. They would find it. And we think this time of year is the early spring, May here on this diagram. So this
David, you
is,
have 2 more minutes.
thank you, Mary. This is work of Kylie Martinod, who estimated deer consumption of honeysuckle leaves and stems from a huge study of browse damage each month at many different sites, scaling up to the landscape. And we also have this pulse in the late summer, which we can't explain right now. And in early spring, honeysuckle's leafed out. Remember that extended leaf phenology. And here's Kylie's data. The percent protein is pretty respectable, 14% on honeysuckle leaves in the early spring. At that time of year, sugar maple and other deciduous trees are leafless, and the twigs have relatively low protein. And so the deer in early spring seeking protein will find these early leafed out shrubs. I'm going to skip over these next couple slides, other than to say honeysuckle comprises a large fraction of the annual deer diet in the Miami natural areas, and may be elevating deer abundance to the extent that that's causing greater browse on native plants. So what are the management implications? It's necessary to manage deer abundance and to manage invasive shrubs. If all you do is one or the other, you're not going to have very much improvement in tree seedlings and other vegetation responses. It's only when you exclude or reduce abundance of deer and remove or reduce abundance of shrubs that you'll see high performance. If managers don't have resources to do both, we recommend first reducing the deer populations and then removing the invasive shrubs. Because if you do it in the other way, first you remove the shrubs, you expose those last few tree seedlings to those hungry deer. The other thing we're exploring is actually to use cut honeysuckle to block deer access. Kevin Lash tried this and had some success just leaving honeysuckle where it falls and planting tree seedlings in the tangle of branches. But what I'm going to try with Callie Kuhn is to make these honeysuckle walls where you suspend a cable between posts and you lean the cut honeysuckle on the cable and clear a rectangle of honeysuckle in the forest, create this rectangle wall of a barrier of honeysuckle, dead honeysuckle. And if that keeps out deer, you have an area of no deer access and no honeysuckle for tree regeneration. Thank you very much.
Thanks, David. And we'll save any questions until after our final presenter. And as a reminder that after our next presenter has finished, we'll have time for questions and answers. So if you have general questions for any of the presenters about woody invasive species, you can type those into the chat bar. And so last we have Mike Schuster, and he will tell us about some native shrubs that can mitigate phenological advantages of invasive buckthorn.
All right, great. Thanks everybody for sticking around. So I'm a postdoc at the University of Minnesota, and this is some work that we've been doing looking at buckthorn phenology relative to some native species in a common environment. So phenology is something that we've heard a lot about, well, at least a little bit about today. So yeah, we'll keep the train going, I guess, if my computer cooperates. There we go. All right, so in general, there's this idea that the process of invasion and restoration goes something like this, where a species is introduced, it establishes on a site, becomes dominant there, and is targeted for removal. And then after that removal happens, the site can be restored. But for buckthorn and for many other invasive species, this isn't really how it plays out in reality. And at least for buckthorn, those removal attempts are often only effective in the short term. And that's because the act of removal actually primes the community for the rapid reestablishment of buckthorn. So if you look at this picture up here, this is a site where buckthorn has been cleared on one side and not on the other. And you can see that those represent very different environments. If we're looking at light availability, for example, on the left, those existing buckthorn shade out any new buckthorn that are coming into the stand. And so you actually have a system that suppresses new buckthorn. But after you remove that understory, there's much more light. And so you move from a system that would suppress buckthorn into one that actually favors its proliferation. So increased light and soil resources mean that resprouts from cut stumps or the extensive seed bank that exists at a lot of these sites can rapidly take over that site once again in only a few years. On top of that, at the same time, a lot of the native species just aren't present in the seed bank anymore. After years of invasion, they're not there to fill the void. And so buckthorn is really the only thing that could take over on that site. And so instead of having that process culminate in restoration, we have a combination of resprouts and germinants pushing us back to earlier stages in this process. This means that stands require repeat treatment within just a few years, and buckthorn removal really becomes more of an issue of routine maintenance and less of part of an active restoration attempt. So this is a site where buckthorn has been removed, and then this photo was taken 2 years later, and all those resprouts have just taken over again. So if part of the problem is that there's a lot of light and really low competition, what if we were able to revegetate by planting in desirable native species in a way that increases competition and lowers light availability? This is something that we do all the time in grasslands. Grassland restoration, generally involves some sort of management activity and then, heavy seeding on top of that. But in forests, it's relatively rarely studied. If even, it's done occasionally in practice, we don't know a lot about it from an academic standpoint. But when it is done, it's done for, either erosion control or biodiversity. You're planting a native species, you could potentially inherently increase biodiversity. Increasing fuel loads to carry fire, particularly in savanna restoration type scenarios, or to provide habitat for wildlife. And seeding can do all of these things in forests, but of course it can also increase competition. So this photo up here is a site where we've seeded a strip of grasses and forbs on the right and haven't done that on the left. And you can see that those are very different competitive environments, right? There's very little vegetation on the left and quite a quite healthy ground layer of grasses on the right. And so these revegetated species can help push that system back from a place where it favors the proliferation of buckthorn into some place where it could either suppress or possibly even exclude buckthorn coming into the site anew. Major challenge to this approach, though, is that, like honeysuckle, buckthorn is often cited as having an extended phenology, meaning that it leaves out early in the spring or holds its leaves late into the fall. And work by Harrington in the 80s and more recently by Jason Fridley have shown critical carbon gains either in the fall or the spring, especially in plants that are in the understory. And this means that the invasibility of a site may be decoupled from its midsummer light availability, or the amount of shading that native trees provide in the middle of summer, and may be more tightly linked to the amount of shade that they offer in the spring or fall. And we looked at this with a study up in Cloquet, Minnesota, where that study is called IDENT. But that study has 196 plots that were established in 2010, comprised of a combination of deciduous and evergreen species that together generate a light and phenology gradient across the experiment. And so 6 years after those trees were planted, we transplanted buckthorn into those plots, so in 2016, and monitored the survival of those buckthorn seedlings and analyzed it respectively to in respect to those different light conditions. And this is what we found. So on the left of each of these, on the y-axis of each of these graphs, we have the probability of survival for the following 3 years, so 2016, 17, and 18, relative to the light availability in the spring, summer, and fall. And what you'll see is that, again, midsummer light availability really didn't have an influence on buckthorn survival. It is relatively insensitive to even extreme shading in the middle of summer. But buckthorn survival declined pretty rapidly once we got to low light availability in the spring or in the fall, and that effect was cumulative over time. So this suggests that from a management perspective, we want to vegetate to minimize buckthorn spring or fall carbon gains. That spring and fall are buckthorn's strength, but they're also its weakness. So if we can target that carbon gain in the spring and fall, perhaps we can outcompete buckthorn. In order to do that, though, we need to know if buckthorn really does have a unique understory phenology. And if not, can we identify which species actually have really high phenological overlap with it? So we did this using an existing ongoing forest restoration experiment using 1.2 by 1.2 meter plots, so a small scale, but dense. And those plots were either planted in with sedges or trees or ferns or a combination of shrubs, or seeded with an herbaceous mix, or just left alone. And those in particular, we're looking at the trees and the shrub plots here. And the shrub plots consisted of red elderberry, common elderberry, American hazel, and gray dogwood. And then the tree plots were a mixture of sugar maple and balsam fir. Since we're curious about phenology, though, and balsam fir is evergreen, we're going to pretend like it doesn't exist and just focus on these 5 deciduous species. So when we monitored these species, we looked at 2 individuals of each species per plot at least once a week, as well as 2 buckthorn that were growing in those plots and 2 additional buckthorn that were in the surrounding area, just in case size made a difference. It didn't, but we still looked at it. In the spring, we started observing when the snow melted and ended when the canopy closed above the plots. At each visit, we assessed whether each individual either had intact buds, had one or more broken buds with some leaf visible, having one or more leaf fully expanded, and then having all their leaves fully expanded. And in the fall, we started doing this when the canopy started to open and ended when there was stable frost or stable snow on the ground. We looked at if they had one or more leaf with visible senescence, so some leaf discoloration, if all their leaves had started to senesce, or if they had lost all their leaves. We did this at 2 sites opportunistically to get 3 sets of observations for each season. So they're not perfect pairs. It was an opportunistic type of monitoring, but we have 3 sets of each spring and each fall. For simplicity, I'm just going to focus on 2 of these phenophases. I'm going to focus on the expansion, where we're looking at how many, looking at the date which species fully leafed out or started to leaf out, and then leaf fall, where they didn't have any leaves remaining, because these kind of bookend the period which these native species could potentially offer shading to incoming buckthorn. As an example, here's our circle pine site in the spring of 2018, looking at, again, leaf expansion date relative to buckthorn. So positive numbers indicate a later leaf expansion date, and negative numbers represent an earlier leaf expansion date for that species. And then for the different species down here on the x-axis, in general, we saw that spring phenology was largely comparable between buckthorn and native species, and that there were relatively few instances where buckthorn actually had earlier spring phenology than those native species. You can kind of see that in this example here, where red elderberry actually had earlier leaf expansion date than buckthorn, but all the other species were statistically identical to buckthorn. This was something fairly common, that most species were the same with buckthorn, but there were cases where elderberry species commonly outpaced buckthorn in the spring. In particular, in 2019, red elderberry outpaced buckthorn by 2 weeks, which corresponded to a pretty significant difference in light availability. Over that 2-week period, the light had changed from being 40% open canopy to a 20% open canopy. And so red elderberry was able to kind of reap that benefit of that extra high light availability over that 2-week period. Again, here's another example from fall, looking at the change in leaf fall date for buckthorn and the other species. Here we saw that actually buckthorn did have an extended phenology compared to some species, particularly dogwood, hazel, and maple senesced much earlier than buckthorn, up to 29 days earlier than buckthorn in some years. However, both elderberry species were generally evenly paced with buckthorn. Red elderberry and common elderberry held their leaves just as long as buckthorn and held them until frost, losing them not to really senescence, but to frost damage and necrosis. And these results echo and support those of a study released earlier this year from Aaron O'Connell and Jessica Savage up in Duluth, where they compared phenology of several different invaders and several different native species, and found that, yeah, in the spring, there really wasn't a difference between natives and invasives, but in the fall, there was. And so why is it then that we have this perception that buckthorn has some special phenology? There's a few different reasons that this could be. Of course, we're only looking at a few sites between both the Connell study and our study here. It's only a few sites, and those are both in Minnesota. So there could be site factors that explain that. We're looking at only a few years here, not decades or anything longer than that. And so there could be some selectivity in our data there. But I think probably a more satisfying explanation is that a lot of times these comparisons are not necessarily made apples to apples. We're comparing buckthorn to the canopy phenology, which it is different. And so when we're comparing buckthorn to canopy trees, we see this difference. Also, when we're comparing buckthorn to other native species, the sites that we look at have just a ton of buckthorn in them. And so it's not readily apparent if there are even other native species there to which we could compare. So just the overwhelming abundance of buckthorns make it stand out and therefore look different, even if it may not be, particularly in the spring. Along these lines, it may also be that communities that lack those species that have comparable phenology are more easily invaded. And so when we see a site that has a lot of buckthorn, it doesn't have those native species in it. And that's why it's heavily invaded, at least in part. And the data from our larger restoration experiments support this. So this is one of our shrub plots. You can see it's largely composed by red elderberry here. And these shrub plots reduced buckthorn survival by 87% after 3 years compared to our unmanipulated control. So you can see that this 3-year survival rate here for our control, and then the different plantings. Trees had a closer survival rate to shrubs, but shrubs still got down to about 6% survival compared to about 50% survival in the unmanipulated control. And so it seems very likely that this trend is driven by red elderberry performance, where red elderberry is highly productive, both in our experiment, but also more generally in the wild. There it forms extremely dense stands and can even be invasive in other parts of the world, like Belarus, for example. Red elderberry phenology beats and matches buckthorn. And especially if we're looking at leaf expansion and leaf drop as being, again, those bookends for shading, we see that red elderberry is offering strong competition for light over the entirety of buckthorn's active period. On top of that, it has these really large compound leaves that can shade quite a lot, right? These aren't small leaves, that it actually puts out quite a lot of leaf tissue. So it seems like red elderberry would be a really strong candidate for revegetation with an eye towards controlling buckthorn. So overall, we saw that buckthorn phenology is different than the canopy, but isn't unique amongst understory plants. Of the 5 species we tested, all of them matched buckthorn phenology in at least some way. But in particular, we saw elderberry species consistently beating or matching buckthorn leaf phenology, especially red elderberry. We also saw extensive mortality of buckthorn under shrub and tree plantings that had high phenological overlap. So this suggests that overall we need to work to operationalize revegetation of red elderberry and other phenologically similar shrubs in order to build long-term biotic resistance into our communities. This way we can potentially avoid the need for repeat management, repeat herbicide usage, and instead build communities that are going to be resistant to buckthorn and likely other species in the long term. So this study was funded by the MIT PC, or MITPIC is a lot easier to say, which is funded by the LCCMR. I also want to thank all of our site partners. We work with a lot of different agencies around Minnesota in order to operate our different experiments. And so they deserve a lot of praise for their patience with us. If you have other questions for me, here's my email as well as our project email. This is just one of many-headed monster that we call the Cover It Up project, including a citizen science project. So if you have questions about that, you can email us at coveredup@umn.edu. Thanks.
Thanks, Mike. And we have a couple of questions specifically for you to get us started. So somebody noted that buckthorn has an allelopathic root system, and if so, does that affect establishment of native shrubs from seed even after buckthorn management?
That's a great question. I haven't seen a lot of strong evidence for the allelopathy of buckthorn. I'd be very excited to read some additional papers on that if there's a recommendation. But in our experience, we haven't seen that there's been a resistance towards seed establishment. So, I have these I have my slides up, so I'm going to use them here, right? So this is a site that is actually extremely heavily invaded. This was done with forestry mowing, so you can see all the mulch remaining there. There's quite a lot of buckthorn and therefore buckthorn root mass at this site, and we still got really good establishment. Granted, this is with a pretty heavy dose of grass seed, but yeah, we haven't seen major limitation based on buckthorn abundance as far as we can tell. Seems to be more tightly controlled by light availability.
Thanks. And I'm curious about the species that you chose for your study. Those are probably specific to certain types of ecosystems, and how could this strategy be used in ecosystems where those species are not maybe native or abundant?
Yeah, so that's another really great question. So, I mean, even these species that we do use are not always the most palatable for land managers, right? If you're looking to restore an oak forest, maybe you're not super excited about planting sugar maple or balsam fir. So I think it's a little bit about, to some extent it's about, do you want the system that you want that might be vulnerable to invasion, or do you want, or are you willing to settle for a different type of community that might be more functional in the long term? So that's part of it, but also, we can look for other species that work in different systems too, right? I mean, elderberries have a pretty wide range, but if we're looking outside of that, there are potentially other species that we can use. Notably, evergreen species are going to automatically have complete phenological overlap. It's just a matter of how quickly they grow and if that's fast enough. So if we can't use these species, I would say look at evergreen species. But I know that that's not everyone's first choice.
Sure. Then we had a comment and question. Somebody says that they've noticed that fire cherry seems to compete relatively well if you do a couple of years of buckthorn suppression, and they're wondering if you have any thoughts on that, and if you think it could be an approach used for landscape-wide management.
I don't have any specific feelings about that species. I will say that cherries, cherries have a lot of similar traits to buckthorn, right? And so you could imagine that would be a useful tool, but I can't say a priori whether or not that species in particular would be an effective control element. It's worth looking at though.
Mm-hmm. And then have you studied allelopathy on other tree species or grasses and how they interact with buckthorn?
So like walnuts, for example. No, we haven't incorporated that into our design, although it's something that we have kept an eye on, or at least an ear to. So we're excited to incorporate additional species into these types of trials, but we haven't made allelopathy a primary focus of this. We're looking more at the light mechanism than the chemical soil pathway.
Sure. Okay. And then David, we have some questions for you as well.
Yeah, let's have everyone bring their cameras back on if you're able to. We could do questions maybe for David and Lauren, and then I'm going to stand in for Theresa, but maybe we'll do those last so that if David and Lauren need to get going, because I can answer some of the Theresa questions. So yeah.
Okay, thanks, Claire.
Awesome.
So someone is wondering how much deer population management is necessary to significantly increase tree seedling recruitment. So for example, do deer have to be extirpated from the landscape in order to see any differences?
That's a great question. There hasn't been that much work on it. There have been all these exclosure studies, that have had very dramatic effects. There's a few papers in the literature on some large-scale deer population management, thinking 7 to 8 deer per square kilometer is low enough to allow some native species recruitment. And probably has to be lower than that to get the preferred species like oaks to regenerate.
And do you know how many deer per kilometer are currently in the area that you've been?
We were working in an area with very high deer density. So there, except for that comparative study with the eight sites that varied in deer abundance. So in an area with a more moderate deer abundance, the effect would not be that dramatic.
Mm-hmm. Okay, and then we had another question. Honeysuckle may protect maple seedlings from browse, as you said, but does a seedling in the shade of honeysuckle extend the time that it takes for the seedling to grow into a sapling?
You know, it probably would. Our experiment was just a 1-year experiment, and then those seedlings were harvested. So that's a good question. The overall performance, so there's deer exclosures. We have data from after 5 years. The only real improvement in sugar maple was the combination of deer exclosure and honeysuckle removal. So there is a honeysuckle shade effect. If all you do is leave the honeysuckles, they'll protect the seedlings to some extent, but you're not going to get good maple recruitment until you manage both honeysuckle and deer.
Mm-hmm. And then to you, Lauren. So lag time seems maybe a bit concerning when trying to prevent introductions of new woody species. Could you remind us of what lag time is? And then do you know what traits might be looked at when considering what woody species may become invasive?
Sure. So lag time sort of refers to when a species is first introduced and you have propagal pressure, which is, the number of introduction events, how many within that introduction event were established. So it takes species some time to establish, to grow, and then to reproduce. And so through time, and so you can have a species, a single individual or a couple of individuals reproduce. After that time until it reproduces, the seed gets to a new area, but all the environmental factors have to be right for that seed to germinate, grow, and reproduce. And so all of that together takes a bit of time. And so species can be referred to as sleeper weeds, which is a species that takes a bit of time to, become abundant enough across the landscape that we recognize that it is an issue. And so a lot of the work that's been done sort of looking at traits of invasive species, both woody plants and also herbaceous plants, can help identify traits that might make certain species less more problematic in the long run. So those species with the small seeds, or for herbaceous species and other species that are wind pollinated, bird dispersed, can make them, raise them to the top of, if we're going to introduce this species, we really need to make sure that we know it's not going to become invasive, as in it's sterile. But then we're seeing things like with Callery pear, that even our best intentions can sometimes go awry. And so all this information helps make us make more informed decisions, but it still means that there's species that were probably introduced 50, 70 years ago that might still be in that lag phase, that might become problematic. And that's where early detection and rapid response, being out there and seeing something that maybe looks a little different, that's pretty abundant locally, and then start talking to your friends and neighbors and your invasive species councils and seeing are people seeing this species. And that I'm actually more from the southeastern United States, and fig buttercup started really popping up in local populations, and it was really the response of the invasive species councils that really started moving efforts to making sure that that species is controlled and mitigated right away. Good question.
And then Claire, I'll pass it off to you if you have any things you would like to ask.
I feel like I probably should address the top ranked question here, which is, why is Ohio so soft when it comes to selling invasive plants, or I guess not regulating them? Okay, so for one thing, definitely stay tuned for the next session, which is really digging into this invasive plants and trade, woody invasive plants and trade issue. For a second thing, I guess from my perspective, Ohio isn't a standout in being particularly soft about it. If I had to pick one standout, and I'm gonna feel like I'm picking on someone, but it would have to be Michigan, because they really do not have, they're not regulating any plants, particularly terrestrial plants and trade. Aquatics, a different story, but it's a different piece of legislation that addresses that. And I think there's a couple of reasons for that. I mean, one is that, and I think the people I know at Michigan Department of Ag and Rural Development would agree with me when I say this. Hopefully wouldn't be offended, but they're a little bit behind other states in terms of evaluating the status and risk posed by invasive terrestrial plants. So that's one thing that's got them behind. And admittedly too, and I don't know the politics behind this, but I do know that of our states that we're looking at, Michigan has the largest horticulture industry in terms of what's being grown in the state. And I would have to imagine the largest lobby as well. So there's that. So Ohio passed this law in, I think it was either 2018 or 2019. It did primarily address species that are already established, and it didn't necessarily get at some of the bigger sellers like burning bush and Japanese barberry, but it did list Callery pear, and that's the only state to have done that. So I don't want to, make it seem like Ohio's regs aren't valuable. All right, so that was that question. I have to hop back and forth between windows, so just give me a second. I know there were some other questions for, I should say too, regarding Ohio, Theresa is actually heads up the Ohio Invasive Plant Council's species assessment division or subcommittee. And so she's always working with folks on assessing plants for Ohio. And that did play a reasonably large part in the regulation, although there wasn't a formal connection there. There was a kind of a behind the scenes awareness of what's going on. There was a question, I can't actually find it because there's a whole bunch, but there was a question about whether crosses of Callery pear have better seedling establishment than seeds from the cultivar. So the true cultivars can't seed. All the cultivars are self-sterile. So they cannot accept pollen from themselves and from other members of their own cultivar. What happens is the cultivars exchange pollen with each other and then bear fruit. And the first generation of fruit are no longer belonging to the cultivar. They're a cross cultivar, or well, not a cultivar at all. They're a hybrid between different varieties. One way you could look at this question though is as Callery pear becomes feral and exchanges more genetic information between several different cultivars in subsequent generations, did they establish better from seed? The answer to that for me would be I'm not sure. they do become more genetically diverse and perhaps more adaptable to different ecosystems, I would say, as genetic material is exchanged. David's probably, or David or Mike are plant biologists, probably you all are. So I don't know if any of you have more details you would add to that. I'm just looking through the questions. So this would be for Mike, and I'm sure the answer will be it depends a little bit, but what types of grasses or sedges would you recommend if someone were to try seeding to help out compete buckthorn?
It depends. Yeah, no, so we've seen the greatest success with limas or wild rye grasses. They make up a pretty big component of our seed mix. Although they tend to do better in a little bit higher light conditions. There's a variety of species that could do well in lower light conditions too. So wild ryes are what we see doing very well.
And just one more question for you. Did your study address common buckthorn, glossy buckthorn, or both?
A critical question, yes. So we look primarily at common buckthorn, Rhamnus cathartica. We're expanding now to consider glossy buckthorn and some other invaders as well. And on that note, I will also amend an earlier response of mine. I did not realize that fire cherry is the same thing as pin cherry, Pyrus pennsylvanica, and we have an experiment looking at pin cherry establishment in the same vein. So we'll find out.
Since you brought it back to pin cherry, just one note on that. I think if you're in Michigan, that species is prohibited from sale because of concerns around fire blight, even though it's native. And I could be wrong, but I've done a lot of digging into these Wraggs, so I think that was one that caught my eye. David, I think I saw in the Zoom chat you have a question for Mike.
Yes, thanks. That was great presentation. All of them were good, but Mike, very good. Two maybe related questions. One, why not look into balsam fir? It's evergreen. Its shading might be really effective. And the other is your team continuing to look at mixtures? Because I could see a mixture of native trees and shrubs might be preferable to a monoculture of red elder.
Yeah, absolutely. So so this experiment that I showed is using mixtures. So the shrubs are in a mixture and the trees are in a mixture. And we actually just this past year have initiated a larger scale tree and shrub planting experiment that is highly diverse. So yes, we are looking at mixtures and hope that kind of niche partitioning will actually be more effective than a monoculture. We'll find out. We're looking at a diversity effect there. And in that experiment, as well as here, we're using balsam fir because it is evergreen and it grows well and could potentially be a good tool in this respect. The why not question is again more of a practitioner question. From a scientific or efficacy standpoint, it seems like balsam fir could be a good tool, but it's just whether or not land managers want to use it. Again, if your goal is to have a particular set of species and we're saying maybe use other species, that those things don't necessarily, you have to make a judgment call. So yeah, thank you.
Well, we are out of time. Thanks everyone for your presentation.